So hi, this is the 10th problem solving session for the course Biochemistry. The course instructor is Professor Swagata Dasgupta from IIT Kharagpur, and I am Shrija Ghosh, a PMRF from IIT Kanpur. Today's session is about carbohydrates, their structures, their stereoisomers and everything. So we'll see how carbohydrates are important for our life forms and how they what are the different structures of carbohydrates and what are the different isomers of carbohydrates, what are the bonds that form between two carbohydrates, and so on. Okay, so we'll discuss few questions regarding that, and feel free to ask me any doubt. Okay, I hope my voice is clear. Please write in the chat box once if there's any problem. Okay, so starting with the first question. Question number 1, identify the correct statements regarding carbohydrates. Identify the correct statement regarding carbohydrates. First statement is they serve as the energy source, fuel and metabolic intermediates. Second is they are majorly found as parts of the structural framework in RNA but not in DNA. Third is they comprise the structural elements of the cell walls of bacteria. And the fourth statement is they are found in the exoskeleton of arthropods. So which of the following statements are correct? Anybody knows the answer? Carbohydrates serve as the energy storage, fuel and metabolic intermediates. They are majorly found as parts of structural framework in RNA but not in DNA. They comprise the structural element of the cell walls of bacteria, and they are found in exoskeleton of arthropods. Okay, we have an answer A, that is 1, 3 and 4. Okay, so 2 is incorrect statement. Yes, you are right. So why second is incorrect statement? Because it is written that the carbohydrates are found as part of the structural framework of RNA but not in DNA. But if you remember, in both RNA and DNA we had sugar moiety, that was in RNA it was ribose and in DNA it was deoxyribose, right? So this is the carbohydrate part of both RNA and DNA. First statement that says carbohydrates serves as energy storage, fuel and metabolic intermediates, that is a correct statement, because carbohydrate is the basic unit of energy. Carbohydrates are basically organic molecules that has carbons and multiple hydroxyl groups, that is OH attached to it. Okay, so that is the basic framework of carbohydrates. So carbohydrates can be written as CH2O N, that is multiple units of CH2Os attached to each other. Okay, then the third statement, they comprise the structural element of cell walls in bacteria. So you will see bacterial cell wall is made of many sugar moieties. There is a material called peptidoglycan. So bacterial cell wall is actually composed of this. This is the basic framework of bacterial cell wall, and peptidoglycan consists of different sugars. All the sugars are basically carbohydrates. Okay, and they are found in exoskeleton of arthropods, that is also true. So carbohydrates make up most of the structural elements, whether it is the cell wall of bacteria, plants, fungi. Most of the structures have carbohydrates in it, and obviously all the energy that we gain from, glucose is the major source from which the metabolic pathways, the bioenergetic pathways start. So when we will study about bioenergetic pathways, you will see how carbohydrates are metabolized in our body to generate energy. So it serves as the fuel for any life form. Okay, so correct answer is A. The second statement is wrong because it also forms the structural framework of DNA, not only in RNA but also in DNA. It forms the structural framework of both RNA and DNA. Okay. Moving on to the next question. Match the carbohydrate with their corresponding structural feature. So we have three carbohydrates here, mannose, fructose and galactose, and we have structural features as C4 epimer of glucose, ketohexose and C2 epimer of glucose. So we have to match the carbohydrate with its structural feature. Anyone wants to answer this? Mannose, fructose and galactose. We have to match it with C4 epimer of glucose, C2 epimer of glucose and ketohexose. So here if you see, among these three sugars only one is a ketose sugar. Both of the other sugars are aldose sugar. So what is an aldose sugar and what is ketose sugar? So aldose and ketose. So aldose means it has an aldehyde group attached to it, and ketose means it has a keto group attached to it. Okay. So mannose and galactose, these are basically aldose sugar, and fructose is a ketose sugar. Fructose has keto group attached to it. So B is 2. Now among mannose and galactose, both are epimers of glucose. What is epimer? Epimer means the epimer is a type of isomer. So your molecular formula will remain same. So what is the molecular formula of glucose? C6H12O6. Okay. So till now you knew that this is the molecular formula of glucose, but for mannose and galactose also the molecular formula will remain same. Only difference will be the difference will be only on single carbon atom. The orientation of the OH will be different. Okay. So here the correct answer is option number C, that is mannose is C2 epimer of glucose, fructose is ketohexose and galactose is C4 epimer of glucose. We will see how. So this is glucose. So you see we have the aldehyde group, the CHO group in all these three. So these are called aldoses. Okay, aldose. And here you have keto group, C double bond O. So this is a ketose. Okay. Now how many carbons are there? We have 1, 2, 3, 4, 5 and 6. 6 carbons are there. So these sugars are called aldohexose. Okay, these sugars are called aldohexose. Why hexose? Because 6 carbon. If it had 5 carbon, it would have been aldopentose, aldotetrose. Like that, we will have the nomenclature. For this, we have 1, 2, 3, 4, 5 and 6. So this is also 6 carbon. That means this is a ketohexose. Okay. This is clear? Aldohexose and ketohexose. Now we will come to epimer. What is epimer? Epimer is basically a single change in the orientation of the side chain on a single carbon. Basically it has 6 carbon. So out of the 6 carbon, if one of the carbon has a different OH orientation, so here you can see in glucose, so all of these are D form of sugars. What is D and L, we will see in the next questions. So basically here you can see on the second carbon the OH is on the right hand side. Okay, here you can see this OH is on the right hand side of the carbon. But in mannose you see that same OH is on the left hand side. Rest the whole structure is same. The third carbon, the fourth carbon, fifth carbon, sixth carbon, everything is same. Only the difference is on the second carbon. The only difference is on the second carbon, that is on the second carbon the OH is on left hand side for mannose. That means it is C2 epimer of glucose. That means the difference is on the C2 second carbon. So C2 epimer of glucose is D-mannose, D-glucose, so D-mannose. Now similarly for galactose it is the C4 epimer. So now you see the fourth carbon, the OH is on the right hand side, but here in galactose the OH is on the left hand side. Okay, the second carbon is same as glucose, the OH is on right hand side. The only difference is in the fourth carbon. All the other carbon, first, second, third, fifth and sixth, all the other carbons are same as glucose. Only difference is in the fourth carbon, that is the OH is on the left hand side, whereas the OH is on the right hand side in case of glucose. That means galactose is called C4 epimer of glucose. Okay, so we have C2 epimer and C4 epimer for glucose. C2 epimer is mannose and C4 epimer is galactose. Okay, this is clear. This part is very important for this chapter. This is the whole basis of carbohydrates. If you don't understand stereoisomers, you have ask me now only, I can explain again. Okay, so this is one type of stereoisomer which is called epimer. Okay, we will see other types in the subsequent questions now. Okay, so I hope this is clear. We will move on to the next question. Question number 3. Just a second. Question number 3. We have 3 structures given, A, B and C. Choose the correct denotation of the above aldohexoses. Now these are aldohexoses. See, 1, 2, 3, 4, 5 and 6 carbons, so, and an aldehyde group. So these are aldohexoses. Now you see the difference between all the carbons and tell me which is glucose, galactose and mannose. Just now I showed you the structure of glucose, galactose and mannose. Now you tell me which one is glucose, which one is galactose and which one is mannose. Which of the following option is correct basically? A, B, C or D? Come on. Just now I showed you the structure, right? So tell me which one is glucose, which one is mannose and which one is galactose? So here you see the center structure, you see it is glucose. Okay, this is glucose, and now you see the second carbon. This is the second carbon. The OH is on the right hand side. Now in this structure you have on the second carbon the OH is on the left hand side. That means this is C2 epimer. Just now I told you this is C2 epimer. That means this is mannose. And in this one the second carbon is same as glucose, third carbon is same as glucose, the fourth carbon is different from glucose. So this is galactose. Okay, so we have A is mannose, B is glucose and C is galactose. So this one is the right answer. So option D. Okay, option D is the correct answer. Now next question is also similar. So now you should be able to answer this. I have already given you the answer. Relation between A, B and C is best denoted by which of the following statements? A is the C2 epimer of B, C is the C4 epimer of B, A is the C4 epimer of B and C is the C2 epimer of B. So which of the following options are correct? Read carefully and try to answer. What is the correct answer? Come on. Yeah, Akshara has answered option A. That is first and second. Yes. So B is glucose here. We discussed right now. B is glucose. So A is a C2 epimer. Here you see on the second carbon the difference is there. So A is the C2 epimer and here you will see on the fourth carbon the difference is there. So this is the C4 epimer. C is the C4 epimer. So this is the correct answer. A is the C2 epimer and C is the C4 epimer of B. Okay. Moving on to the next question. Structural representation of cellulose is shown below. Choose the correct answer. The bond joining the monosaccharide unit is. What is this bond that is joining the two monosaccharide units? So this is one monosaccharide and this is another monosaccharide. And what is this bond here? And this is the structure of cellulose. What would be the correct answer? Okay, Akshara is saying option D. That is beta 1,4 glycosidic linkages. Okay, so that is the correct answer. But how do we determine whether it is a beta or alpha linkage, and how are we doing the numbering? So numbering will start from when glucose units are written in cyclic form. We will start the numbering from the carbon next to the oxygen. So this is 1, this is 2, this is 3, this is 4, this is 5 and this is 6. Okay, this carbon is 6th carbon. Now you see here, so the and now I will number this also. So 1, 2, 3, 4, 5 and 6. Okay. So now you see the bond is between 1 and 4. So 1, 6, the options that has 1, 6 are already eliminated. Now we have 1. Okay, so now we have 1, 4 and only one option. But what is the difference between alpha and beta linkage? So you see this oxygen here, we have the hydrogen towards downward side and the oxygen is on the upward side. And you see the 6th carbon. This 6th carbon is also on the same side. So if the oxygen and the 6th carbon is on the same side, that means it is beta. Okay. And if it was opposite, so let's say I had, a structure, a glucose molecule, so CH2OH, and I had the OH here, H here. Rest of the structure will be same. Okay, rest of the structure will be same, only difference is on the first carbon. Okay, now the OH is opposite in direction as compared to CH2OH. CH2OH is facing upwards and OH is facing downwards. That means the orientation are opposite, This opposite orientation is called trans orientation, and this glucose is alpha glucose. Okay, whereas in this the orientation is same, and the same orientation is called cis orientation, and it is beta glucose. Okay, so this is, since the linkage is starting from a beta glucose, this is beta, and the linkage is between 1 and 4 carbon, so beta 1 4, and I have told you in the previous session also, any bond between sugars is called a glycosidic bond. Glycosidic bond means a bond that is forming between sugar molecules. So this is a bond between two sugar molecules, so this is a glycosidic bond between the first and the fourth carbon, and the first molecule that is forming the bond is a beta molecule, so beta 1,4 glycosidic linkage. Okay, beta 1,4 glycosidic bond is the correct answer. Okay, I hope this is clear. In case of any doubt, you can write in the chat box, or you can unmute your mic and ask. Okay, moving on to the next question. Here we have different monosaccharides and their characteristics or usage listed, and we have to match the monosaccharide column to its corresponding characteristic or usage column. Okay, so we have D-glyceraldehyde, D-glucose, D-fructose, D-galactose and D-ribose, and characteristics are used in RNA, part of milk sugar, sweetest of all sugars, most important in diet, and simplest sugar. So now we have to match glyceraldehyde, glucose, fructose, galactose and ribose. Tell me what would be the correct answer. Read carefully and try to match. Okay, I'll help you. So first is glyceraldehyde. Glyceraldehyde is the simplest sugar. Okay, so one will be E. So glyceraldehyde is basically a 3 carbon molecule. So we have CHO, we have CHOH and then CH2OH. So this is glyceraldehyde, the 3 carbon sugar. This is also known as aldotriose. Okay, so it's an aldehyde, and it is, so you remember the structure of glycerol, right? So we had 3 carbon when we were doing when we were studying the structure of fatty acids. You remember, from the glycerol molecule only the fatty acids were derived. 2 had alkyl chains attached and 1 made the polar head group, and that's how we had the amphipathic molecule with a polar and a non-polar part, right? So similarly, carbohydrates are also derived from such structures. So this is glycerol. In this glycerol, when this group is converted into aldehyde, we get, what do we get? We get glyceraldehyde. So this is the simplest sugar. Okay, this is the smallest sugar possible with 3 carbons. So one is E. Now we have glucose. Glucose is most important in the diet, because I told you earlier also, in all the bioenergetic pathways, when we will study the bioenergetics, you will see how important glucose molecule is, and any other sugar is first broken down into glucose, and then it enters the bioenergetic pathways for metabolism, for energy generation. Okay, next is fructose. So fructose is the sweetest of all sugar. Fructose is the sweetest of all sugar. It is the sweetest sugar available. Then we have galactose. Galactose is generally found in milk sugar, so it is a part of milk sugar. And fifth is ribose. You all know, I have told you multiple times that ribose is part of RNA. So we have two different sugars in nucleic acid, ribose and deoxyribose. Ribose is part of RNA, deoxyribose is part of DNA. Okay. So we have, the correct answer is option B. Option B, that is first E, glyceraldehyde, simplest sugar, glucose, most important in diet, fructose, sweetest of all sugar, galactose, part of milk sugar, and ribose used in RNA. Okay. So this is the structure. See here, this is glyceraldehyde, then we have glucose. Glucose is a 6 carbon sugar, 1, 2, 3, 4, 5 and 6. What will we call this? We will call this aldohexose. This is called aldotriose. Why? Because we have an aldehyde group, so aldo, and triose because 3 carbons are there. This will be, these two will be aldohexose. Why? Because aldehyde group is there and 6 carbons are there. Now these this is fructose, that is called ketohexose. Okay, ketohexose. Why? Because ketone group is there and 6 carbon is there, 1, 2, 3, 4, 5 and 6. Okay, and this is ribose, this has 5 carbons, 1, 2, 3, 4 and 5. So this is called aldopentose. Okay, aldopentose, that means an aldehyde group is there and 5 carbons are there. So ribose is an aldopentose, glucose is an aldohexose, glyceraldehyde, that is the simplest sugar, is aldotriose. Then we have ketohexose, a ketone sugar, ketose, that is fructose. Okay, this is clear. Moving on to the next question, question number 7. Which among the following statement is or are false about mutarotation? Okay. First statement is mutarotation is the interconversion of different anomers. Second is the beta anomer of D-glucose is less stable because of the equatorial position of C1 hydroxyl group. Third statement is interconversion occurs due to transient form of open chain form. And fourth is relation between the two anomers are determined by their relative stability. So which of the following statement is false about mutarotation? You have to identify the incorrect statement. Okay. Tell me what would be the answer. Which statement is incorrect about mutarotation? The correct answer is option. Okay, Akshara has answered option D. That is, fourth statement is incorrect. Okay, fourth statement is actually correct. The incorrect incorrect statement is the second statement. The beta anomer of D-glucose is less stable. It is actually more stable. So beta anomer of D-glucose is more stable because of the equatorial position of C1 hydroxyl group. So what is mutarotation? Mutarotation is basically when the cyclization of this open chain form happens. The cyclization of this open chain form can lead to two anomers of glucose. Okay. So when the cyclization is happening, so cyclization happens between the first and the fifth carbon. Okay, the lone pairs of the hydroxyl of the fifth carbon attacks the first carbon, and that's how the cyclization happens. So this is the first carbon, second, third, fourth, fifth. So this, the fifth carbon had the OH that attacked the first carbon and cyclized in this form, and we have the sixth carbon attached to the fifth carbon. Okay. So while this cyclization, this OH can go either in the axial position or on the equatorial position. That is, this OH can be opposite to this sixth carbon. So here we have 1, 2, 3, 4, 5 and we have 6 carbons. Okay. So this OH on the first carbon, this OH on the first carbon, this can be opposite to the sixth carbon, that will make it alpha glucose, because this is in trans, the OH is in trans position with respect to the sixth carbon, CH2OH. Here 1, 2, 3, 4, 5 and 6. So the OH is in the same direction as CH2OH. So this is cis orientation. The OH is in cis orientation with respect to the CH2OH group, and this is called beta glucose. Now these two are basically 6-membered ring structure, right? These two are 6-membered ring structure, so they can take up a chair conformation. When they are taking a chair conformation, now all the carbons are sp2 hybridized, right? So they will have a tetrahedral geometry, that is something like this. Okay. So a carbon with 4 atoms attached, so that will make a tetrahedral. So this carbon is in tetrahedral geometry, this carbon, every carbon is in a tetrahedral geometry. So this is 1, 2, 3, 4, 5 and 6. Now you see, in each case you will have an axial bond and an equatorial bond, right? The axial bond that is going on top, or like that is going through the plane, either above the plane or below the plane, and the equatorial bond. Okay. So each atom has an axial bond and an equatorial bond. So in the alpha glucose, since this CH2OH, this is the CH2OH, right? CH2OH, this group is on the equatorial bond, right? So alpha glucose, the OH is an axial bond, that's how it is opposite. CH2OH is on the equatorial bond and OH is on axial bond. So this is alpha glucose. Whereas in case of beta glucose, both CH2OH and the OH of the first carbon both are on equatorial bond. So this is on equatorial bond, this is the axial bond, this is the equatorial bond. Okay, this is the equatorial bond, and here the hydrogen will be on axial bond. Okay. So when the OH is on the equatorial bond, this structure becomes more stable. This structure becomes more stable because there is less steric hindrance. Okay. In this case, the structure is less stable. Okay. So beta glucose, this anomer is more stable. So these two are called anomers. So we studied about epimers where there was change on one carbon. Now in case of anomer, anomer forms when the cyclization of glucose is happening and there is a difference in the orientation of one carbon with respect to another carbon. So the orientation of the first carbon, the side chain of first carbon with respect to the sixth carbon, if it is trans, then it will be alpha glucose, if it is cis, then it will be a beta glucose. Okay. So this is called anomers. And this rotation about the axis. So when this when a reaction with glucose is happening, there is a chance that glucose can go through this open chain. This is the open chain conformation. Okay, open chain conformation. So there is a chance that glucose can go into an open chain conformation and then again cyclize. When the recyclization is happening, there is a chance that this carbon has rotated. Okay, this carbon has rotated. So that means there is a chance that from alpha glucose the molecule has rotated into another anomeric form, that is beta glucose. So, this phenomena is called mutarotation. Okay. So, rotation of one molecule, one anomer into another through a transition state of open chain form. So, here we have mutarotation is interconversion of different anomers, this is true. Then interconversion occurs due to transient form of open chain form, this is also true. Relation between two anomers are determined by relative stability. So, how much percentage of alpha and beta glucose will be present in a solution, that will be dependent on the stability. Since beta is more stable, beta will be in higher percentage as compared to alpha. Okay. So, beta is more stable. So, this is the false statement actually. So, correct answer is option B. This is clear. What is mutarotation, that part is clear, and what is alpha and beta form of glucose, at least this thing should be clear by now. You can ask if you have any doubt. Okay, then we will move to the next question, question number 8. In aqueous solutions, D-glucose exists as either alpha D-glucose or beta D-glucose. This occurs because option A, aldehyde reacts with alcohols to form hemiacetal. Option B, aldehydes are stereoisomers of alcohols. Option C, glucose contains a stereocenter, and option D, glucose exists in the aldopentose form. So, what would be the correct answer here? What would be the correct answer here?
900 രൂപ 18 നായിട്ട് 36 എന്ന് പറയുമ്പോഴേക്കും 10.
So, in aqueous solution, D-glucose exists as either alpha D-glucose or beta D-glucose. This occurs because, what is the reason? Just now we discussed about mutarotation. So, this is asking that only, why the mutarotation is happening. I explained mutarotation just now. Why the mutarotation was happening? Because of the mutarotation was happening because of cyclization, right? So, mutarotation was happening because of cyclization. The cyclization was happening between which two groups? Between the alcohol and the aldehyde group. So, aldehydes react with alcohol to form hemiacetal. So, that is the correct answer here. Option A, aldehydes can react with alcohols to form hemiacetal. So, if we have an aldehyde and we react it with alcohol, so the oxygen, the lone pair of oxygen can attack this carbon. In turn, this oxygen can pick up a proton and the OR of this alcohol can join. So, basically if we have R1 CHO and if we have an alcohol OH with R2. So, this oxygen will attack this carbon and this oxygen can pick up another proton. So, it will become basically, this oxygen has picked up a proton. So, this becomes OH, the H will already be there and this oxygen has attacked this carbon. So, this oxygen will come along with the R2. Okay, so this is a hemiacetal formation. So this is a hemiacetal formation. Okay. So in this molecule we have both aldehyde and hydroxyl groups, like alcohol and aldehyde groups, right? So the cyclization can happen within the molecule. So we have 6 carbon molecule. If I write it in this way, we have this aldehyde group, the first carbon, then the second carbon CHOH, then the third carbon with CHOH, fourth carbon CHOH, fifth carbon with CH, and this is the OH. So CH and OH, and the sixth carbon, that is CH2OH. Okay. Now the OH of the fifth carbon can come close to the aldehyde group of that is on the first carbon, and it can attack the first carbon, and in turn the proton can be acquired by this oxygen and the cyclization can occur. So this is basically the oxygen of the fifth carbon. So this oxygen is basically here, and we have the cyclized compound. So we have 1, 2, 3, 4, 5 and 6 carbon molecule in a cyclic form. So this is the cyclization. So this is the hemiacetal formation. This is the hemiacetal formation between an aldehyde and an alcohol group within the molecule of glucose. And in this, once this cyclization happen, now the mutarotation can happen. So if this OH is opposite to this CH2OH, this is alpha form. So this, the one that is drawn here, it is alpha form. If I ask you to make the beta form, it will be like, how will we make the beta form? So CH2OH is here. So this OH will also be on the upper side, right? Okay. So OH and CH2OH are on the same side. So this is beta form and this is alpha form. Okay. So that's how the cyclization of a glucose molecule occurs. So this is the open chain form, or this is the open chain form in a 3-dimensional structure. This is the open chain form and this is the cyclic form. Clear? This is clear? So basically the in aqueous solution the D-glucose can exist as either alpha or beta. So this is the alpha, this is the beta. So D-glucose can occur in both form, alpha and beta form, and this is happening because of the rapid cyclization and the rotation of the carbon along the first carbon, the rotation of the hydroxyl group of the first carbon along across the bond axis. Okay. So OH can be cis to the sixth carbon, or OH can be trans to the sixth carbon, and it can determine whether it is an alpha glucose or beta glucose. Okay. Okay, moving on to the next question, question number 9. Different stereoisomers formed due to cyclization of aldehyde or ketone carbon are called. Option A epimer, option B anomer, option C constitutional isomers, and option D enantiomers. So what is it called? Just now we discussed about the cyclic isomers. So what are those isomers called? Epimer, anomer, constitutional isomers, or enantiomers? What would be the correct answer? Come on. We have discussed this. We have discussed epimers. We have discussed anomers. We have discussed enantiomers. So what is it? when the cyclization is happening, what is forming? So if let's say glucose is cyclized in an aqueous solution, that means the glucose can have two isomers, alpha and beta. What are alpha and beta? What are alpha and beta? They are basically anomers. Okay, the correct answer is B, anomers. So I have told you about epimers. What was epimer? Epimer was basically a change on a single carbon. So that was epimer. So glucose, mannose and galactose, they were epimers. Anomers is basically once the cyclization has happened due to hemiacetal formation between aldehyde and alcohol, it can form two anomers, alpha and beta, based on the orientation of the hydroxyl group as with respect to the sixth carbon. Okay, so this is anomers. Now which of the following is an anomeric pair? Now you can answer this. Now you can answer this easily, right? Which of the following is an anomeric pair? Here I have told you alpha and beta, but the thing that you have to look into is this is D glucose and D mannose. So glucose and mannose are epimers. So this is not an anomeric pair. Glucose and glucose, alpha and beta, but the difference is here L and D. So this is not an anomeric pair. In the anomeric pair, the enantiomer isomer should be same. So alpha D glucose and beta D glucose, this will be an anomeric pair. Okay, so this will be the correct answer. Alpha D glucose and alpha L glucose, again this is the difference between D and L. So this is not an anomeric pair, this is an enantiomeric pair. Okay, so this is an epimer, this is an anomer and this is an enantiomer. Okay, so correct answer is option C. So here we can see the difference between D and L. So this is called enantiomer. Enantiomers. So in D glucose, so if I number the carbon 1, 2, 3, 4, 5 and 6, the chiral carbon that is farthest from the most oxidized carbon. So most oxidized carbon is this aldehyde carbon. So first carbon is aldehyde carbon. So farthest chiral carbon. So what are the chiral carbon here? We have this as the chiral carbon, this as the chiral carbon, this is the chiral carbon and this is the chiral carbon, right? So the farthest chiral carbon, fifth carbon is the farthest chiral carbon. Now in this carbon you see the OH. If the OH is on the right hand side, we will have a D glucose. If the OH is on the left hand side, we will have L glucose. So this enantiomer is also called mirror image. So if I keep a mirror here, you can see this is exactly a mirror image of the of this molecule. So if I superimpose this molecule, if I superimpose this molecule, what will happen? So it's like our two hands. I have two hands, these are mirror image. If I superimpose, it fits over each other, right? Similarly, if I take the D and L glucose and overlap each other, superimpose D glucose with L glucose, it will perfectly fit. Okay. So do you understand this? So these are mirror images. We have the D glucose here, we have the L glucose here. If I superimpose, it will perfectly fit. Okay. Now if I am superimposing it like this won't fit perfectly, right? So that means the isomers that are mirror images of each other, they are called enantiomers. Okay. The isomers that are not the mirror images, they are called diastereomers. Okay. So we have another term called diastereomers. Okay. So this is not a mirror image, this is a mirror image of each other. Okay. So D and L glucose are called enantiomers, which is basically a mirror image. Okay. Now I have already talked about alpha and beta glucose, so this is called anomer, and I have talked about epimers where the change is on a single carbon, so these are epimers. So these all are basically types of stereoisomers. Okay, types of stereoisomers. What is an isomer? Isomer means the molecular formula is same. So all these molecules, D-glucose, L-glucose, alpha-glucose, beta-glucose, mannose, galactose, everything, for everything the molecular formula is C6H12O6. You count the carbon atoms, 6 carbons are there. You count the oxygen, 1, 2, 3, 4, 5, 6. 6 oxygen is there in mannose, 6 oxygen is there in glucose. Okay, so and if you count the hydrogen, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12. So all the molecules shown on this slide, every molecule has the molecular formula C6H12O6. But there is a difference in the structure, there is a difference in their geometry, there is a difference in their spatial arrangement. So that difference is basically what makes different stereoisomers, and that difference will only arise if there is an asymmetric carbon. Okay, so this is an asymmetric carbon, means four different groups are attached to this carbon. Similarly, this is an asymmetric carbon, this is an asymmetric carbon. So all the asymmetric carbons can have now two types of geometry. Okay, each asymmetric carbon can have two types of geometry, basically a mirror image. So if you see only this second carbon, only this carbon, if you match, this is a mirror image, right? Either the OH can be on the left or OH can be on the right, or OH can be on the left. Okay, but the whole molecule is not a mirror image, that's why they are not enantiomers, they are epimers. In case of enantiomers, the whole molecule is a mirror image. Okay, if you compare the fourth carbon here, it is a mirror image, but the whole molecule is not a mirror image. Okay, so each chiral carbon can have two different orientation, and thus number of chiral carbons, more the number of chiral carbons, more the number of stereoisomers we can get. This is clear? Okay, we will move on to the next question now. Question number 11. Starch and glycogen are both polymers of option A fructose, option B sucrose, option C alpha D glucose, option D beta D glucose. Starch and glycogen are polymers of. Anybody knows the answer? Starch and glycogen are polymers of fructose, sucrose, alpha D glucose and beta D glucose. What would be the right answer? Come on, what would be the right answer here? Starch and glycogen are, they are the polymers of alpha D glucose. So option C is the correct answer here. Okay, so this is the monomeric unit of starch. So you see this particular glucose, tell me whether this is alpha or beta. You see the CH2OH is towards top, so I will number this 1, 2, 3, 4, 5 and 6. So CH2OH, the sixth carbon is on the top and the oxygen is towards the bottom. So this is trans, that means this is alpha. Okay, here also you see the linkage. So I'll number this 1, 2, 3, 4, 5 and 6. CH2OH is towards top and O is towards the bottom. So this is also an alpha glucose. Okay, so amylose and amylopectin are 2 units of starch. This is a linear form and this is the branched form. So this linkage you see here, so this is between the first and the fourth carbon, right? 1, 2, 3, 4, 5 and 6. So this is linkage between first and fourth carbon. So this is called alpha 1,4 linkage, whereas this is the linkage between first and sixth carbon. So 1, 2, 3, 4, 5 and 6. So first and sixth carbon. So this is the alpha 1,6 linkage. In glycogen you see, you have here first and this is the sixth. So CH2 is towards top and O is towards the bottom. So this is also alpha. Okay, this is the monomeric unit and this is an alpha glucose. Now you see the linkage. This is between 1 and 4. So this is alpha 1,4. This is alpha 1,4 and this is alpha 1,6. Okay. So both starch and glycogen have alpha 1,4 and alpha 1,6 linkages of alpha glucose. Okay. This is clear? So what kind of linkages are these? What kind of linkages are these? These are glycosidic linkage. These are glycosidic linkages. Okay. Why glycosidic? Because the linkages are between two sugar molecules. So this is glycosidic. Okay. So linkage is between two glucose molecules. What type of glucose molecules? Alpha glucose molecules. Okay. Identify the conformations A and B of the cyclohexane backbone. State which conformer is more stable and why. So we have two forms here. Tell me whether A is chair or boat or B is chair or boat, and tell me which one is more stable and why. Okay. So the correct answer here is option B. A is boat and B is chair. So you see it looks like a boat, right? It looks like a boat here. So this is called a boat conformation and this is called a chair conformation. Why a chair? So this, if you imagine, these three carbons on one plane and these three carbon on the next plane, so we have 1, 2, 3, 4, 5 and 6. 6 carbons are there. So 1, 3 and 5. If you see 1, 3 and 5. 1, 3 and 5 are on same plane on the top part, and 2, 4 and 6. 2, 4 and 6 are on the bottom plane. Okay. Imagine a chair where the legs are basically 2, 4 and 6 and the top part is made by 1, 3 and 5. So this is a chair form and this is a boat form. So chair form is actually more stable than boat form. So B is more stable than A. So this is boat and this is chair, and B is more stable than A. Why? Because of less steric crowding. What is steric crowding? Basically, just a second. What is steric crowding? Steric crowding basically means you have two bulky groups. If those two bulky groups are on the same side, they will have steric hindrance. They can't fit on the same plane. Okay. So, if those two bulky groups are on the opposite orientation, it would be easier for the molecule to be stable on a space, like in the space. Okay. So, two molecules, if they want to fit in the same space, it would be difficult. They will hinder each other's spaces, right? So, if they are on the opposite direction, so both of them will have enough space to be stable in that particular geometry. Okay, so that is steric crowding, and chair conformation has less steric crowding than boat conformation. Okay. So, the next question is question number 13. Carbohydrates that are optically active, having the ability to rotate the plane of polarized light, light rotated in the clockwise direction is generally represented by the symbol. So, carbohydrates that are optically active, they have the ability to rotate the plane polarized light. Okay. Now, the light rotated in the clockwise direction is represented by which symbol? So, whether it is represented by L, R, plus or alpha. Okay, we have an answer, alpha symbol. Okay. Anyone else wants to answer? The correct answer is actually plus. So, the plus symbol is actually correct. So, here we have L, R, alpha and plus. So, all four represent different properties about rotation. So, we have L and D form, L and R form. L means levorotatory. So, that is the form of stereoisomer where the isomer can rotate the plane polarized light towards the left hand side. So, we have the plane polarized light, if it is rotated towards left hand side, it is called L. The R means rectus in Latin and it means right. So, molecules that rotate the plane polarized light to the right hand side, they are called R isomers. Okay. So, we have RS nomenclature, we have DL nomenclature. So, there are different types of nomenclature in stereochemistry. So, different stereoisomers can be named based on D and L or based on R and S, depending on whether the plane polarized light is rotated towards left or right. Okay. Now, come to plus and minus. So, plus and minus comes when it refers to the direction in which the pure enantiomers of this compound rotate the plane polarized light either in the clockwise direction or in the anti-clockwise direction. Okay. So, if it is rotating the light in the clockwise direction, it is indicated by plus. If it is rotating the light in anti-clockwise or counterclockwise direction, it will be rotated as minus. Then comes another nomenclature where we use alpha and beta, that I have already explained. So, alpha, the name given to the configuration of a cyclic sugar where the oxygen on the anomeric carbon is on the opposite face of the ring relative to the substituent on the other carbon flanking the ring oxygen. So, basically the OH is either opposite to the carbon that is flanking the ring system. So, that in that case the nomenclature will be alpha. If the OH is in the same direction as the carbon that is flanking the ring system, it will be beta, where the two substituents are on the same faces of the ring. Okay, opposite faces of the ring, that means alpha, same face of the ring, that will be beta. Rotation of plane polarized light in a clockwise direction will be denoted by plus, in a counterclockwise direction will be denoted by minus. Rotation of plane polarized light towards left will be denoted as L, or in some cases will be denoted as S, and rotation in the right direction of plane polarized light will be denoted as D, or in some cases R. Okay, so DL nomenclature, RS nomenclature, alpha beta nomenclature, and plus minus nomenclature. These are 4 different nomenclature in stereochemistry. Okay. Okay. So now we move to the next question. Question number 14. Main component of the plant cell wall is. So I have told you all the structural forms that we see, whether it is bacteria, plant, animal, everything is composed of carbohydrates. So main component of plant cell wall is also a carbohydrate. Which of the following is that? Option A, cellulose, option B, glycogen, option C, peptidoglycan, and option D, lignin. Tell me what would be the correct answer. Okay, Akshara has answered cellulose. So the main component of plant cell wall, yes, it is cellulose. So we have here cellulose, glycogen, peptidoglycan, and lignin. So glycogen is actually a storage molecule. So this is a carbohydrate that acts as a storage molecule. Where it acts as a storage molecule? In animals. Okay. Peptidoglycan is also found in cell wall, but it is found in the cell wall of bacteria. Okay. So cellulose is found in cell wall of plant. Cell wall of plants, that is cellulose. Cell wall of bacteria, peptidoglycan. Lignin is found in cell wall of fungus. Okay. Okay. So cellulose is the correct answer. So this is the structure of cellulose. Tell me what kind of linkage is this? Whether it is alpha or beta, and what would be the numbering? Tell me, I have told you about alpha and beta linkages. So tell me how to, what would be the nomenclature of this particular linkage marked here? This is the structure of cellulose. Okay, just identify whether it is an alpha linkage or beta linkage. Come on, look at the oxygen, look at the 6th carbon. If they are on the same side or if they are on the opposite side. If you guys have still not understood what alpha and beta linkage is, please ask, otherwise answer this question. There is no point in having these problem solving sessions if you are not going to solve your problems clearly. So what is this linkage? This is a beta linkage. See here, the oxygen is on the top, so is the 6th carbon. So this is 1, 2, 3, 4, 5 and 6. So 6th carbon, the oxygen is also on the same side as the 6th carbon. So this is a beta molecule, and the linkage is between 1st and the 4th carbon, 1, 2, 3 and 4. So this is a beta 1,4 glycosidic linkage. Okay. Okay, this is a beta 1,4 glycosidic linkage. So cellulose has a beta 1,4 glycosidic linkage. Moving to the next question, question number 15. Identify the product P obtained in the scheme shown. Name the reagent used to obtain P. which can also serve as the test for reducing sugars. So you have to identify the product P. So we have here beta D glucose, and it is written in the open Fischer configuration. So we have the D glucose, which is converted into the molecule P, where the aldehyde group has been converted into an carboxylic group, and the Cu2 plus is reduced to Cu plus. It is a test for reducing sugar. So what is the reagent used to obtain P, and what is the test called? What is P? Identify this product P, and what is the reagent that is used to convert this D glucose into this P? Options are P is glucopyranose and the reagent is Felling's reagent. Option B, P is D-gluconate and reagent is Edmond degradation. Option C, P is glucopyranose and reagent is Edmond degradation. And option D, P is D-gluconate and the reagent is Felling's reagent. What would be the correct answer? Which option? A, B, C and D? So here the conversion is basically glucose is reducing this copper Cu2 plus into Cu plus formation. See, from 2 it went to 1 plus, right? So copper is reduced here, and what is reducing this copper? Glucose. So glucose is called a reducing sugar. It is called a reducing sugar. Why? Because it can reduce these molecules. So it can reduce the Cu2 plus into Cu plus. In turn, it is getting oxidized into molecule P. Molecule P is nothing but gluconate. So D-gluconate. Sorry. So molecule P is D-gluconate and the test is Felling's reagent. So the correct answer is option D. So Felling's reagent is a reagent that can be used to detect reducing sugar in any solution. So if I give you a solution, unknown solution, and I ask you to tell me whether it is a reducing sugar or a non-reducing sugar. Reducing sugar means it has a free aldehyde or a ketone group. Okay, non-reducing sugar will not have a free aldehyde or a ketone group. So if I ask you whether it is a reducing sugar or a non-reducing sugar, you will do a Felling's test. You will use the Felling's reagent, which will basically have the Cu2 plus and Cu plus, and if it is reducing from Cu2 plus to Cu plus, that means you will have a reducing sugar in the given solution. How do we detect this Cu2 plus and Cu plus? So basically Felling's reagent has a blue color. So Cu2 plus is blue in color. So this is the reaction that is happening. So any reducing sugar, this can reduce the Cu2 plus into Cu plus, that is Cu2O. Now cuprous oxide has a red color. So there will be a distinct color change from blue to red. So once I have the Felling solution that is blue in color, I have in another test tube a sugar solution that is transparent, and I don't know whether it is a reducing sugar or a non-reducing sugar. Okay. So I will take the Felling's reagent and add to the reducing sugar. If the color changes from blue to red, that means the sugar that was given was reducing sugar. If the color does not change, that means it was a non-reducing sugar. Okay. So that is a very famous test for detecting reducing sugar. Okay. So Felling's reagent, basically that comprises of Cu2 plus ions. They are reduced by the reducing sugar to form Cu plus ions or Cu2O, as we know, and that is red in color. So there will be a distinct color change from blue to red, and that color change can tell you whether it is a reducing sugar or not. And since this sugar is reducing this, so it will in turn oxidize. So in any redox reaction, you know, if one thing is getting reduced, another thing will get oxidized. So CHO will oxidized into C double bond, COO minus, that is a carboxylate ion. So aldehyde is getting oxidized into carboxylate ion. So that means if we have an aldose sugar, it will become a carboxylate sugar. That means if we have glucose, we write aldose, like sugars, as O's with the suffix, right? So we have glucose, so once it is converted into carboxylate, it will be called gluconate or gluconic acid. Okay, so if I add a proton to this oxygen, it will become gluconic acid, or if it is an ionic form, it will be gluconate. Okay. Okay, so the answer is, the molecule P is D-gluconate. Since we started with D-glucose, we will get D-gluconate. Here you can see the OH is on the right hand side. So D-gluconate, and the reagent is Felling's reagent. This is clear? This is clear to everyone? Okay. Question number 16. A is a B form of glucose found in C. So statement 1 is starch. So starch is a polymeric form of glucose found in plants. Statement 2 is starch is a monomeric form of glucose found in animals. Statement 3 is glycogen is a polymeric form of glucose found in animals. And fourth statement is glycogen is a monomeric form of glucose found in plants. So which statements are correct? 1, 2, 3 and 4. Which statements are correct here? Starch is a polymeric form of glucose found in plant, or starch is a monomeric form of glucose found in animals? Glycogen is a polymeric form of glucose found in animals, or glycogen is a monomeric form of glucose found in plants? Which statements are correct? Okay, we have the answer option D, that is first and third statement. Yes, very good. So starch is a polymeric form of glucose that is found in plants. So I've told you starch and glycogen, these are two storage molecules. They are the storage molecules. Okay, so in plants the storage molecule is starch, in animals the storage molecule is glycogen. Both are polymeric forms of glucose. So basically, if our body wants to store energy, they will store the carbs in the form of glycogen in case of animals, and plants will store the carbohydrates in form of starch. Okay, and both are basically polymeric forms of glucose. So first and third statement is correct. So I have shown you the structure of starch and glycogen in the previous slide. So starch had alpha 1,4 and alpha 1,6 linkage, so did glycogen. So both starch and glycogen are polymeric forms of alpha glucose. Okay, that is also important to remember. It is polymeric form of alpha glucose. Okay, the next question, question number 17. This is clear till now? Any doubt? If you have any doubt in any of the questions, please ask. Now we are moving to next question, that is question number 17. Which of the following monosaccharides is the majority found in human body? Which of the following monosaccharides is the major monosaccharide basically found in human body. The D type, the L type, the D and L type both, and none of the above. So which is majorly found? Yes, so we have the answer option A. Akshara has answered option A, that is D type. Yes, that is the correct answer. So in human body, most of the monosaccharides that is present is in the D form. So D glucose. That's why if you have seen all the questions, in all the questions we had D glucose, D mannose, D galactose, because the D form of sugar is majorly found in human body. In case of amino acids, if you remember, the L form of amino acid was major and D form was unusual in human body. Mainly L form of amino acid was found. Similarly, for sugars, for carbohydrates or monosaccharides, D form of monosaccharides is more common in human body than L form. L form is rarely found or not found in human body. Okay. So option A is the correct answer. Okay, the next question, question number 18. The total number of chiral carbon in glucose and fructose are. So we have seen the structure of both glucose and fructose. What do you mean by chiral carbon? Chiral carbon means the carbon, the asymmetric carbon, that is the carbon that is attached to 4 different atoms in a spatial plane. So how many chiral carbons are there in glucose and how many chiral carbons are there in fructose? Option A, 4 in each. Option B, 3 in each. Option C, 4 in glucose, 3 in fructose. Option D, 3 in glucose, 4 in fructose. What would be the correct answer? Think of the both the structures. Think of glucose and fructose and then try to answer. What would be the number of chiral carbon in glucose and what would be the number of chiral carbon in fructose? Okay, Akshara says 4 in both. Okay. So, if you remember, I have told you glucose is a aldohexose and fructose is a ketohexose, right? So, this has the aldehyde group and this has the keto group, right? Now, glucose has 4 chiral carbon, that is correct, but fructose has 3 chiral carbon. How? We will see now. So, this is glucose. So, now if I ask you asymmetric carbon, so you see the first carbon is attached to 2 hydrogens. So, it is not asymmetric because it is attached to 2 same molecules, right? If I just draw the first carbon and write this as R group, this is not asymmetric because two of the 4 atoms are same. So, this won't be a chiral carbon. Okay, this won't be a chiral carbon. Now, coming to the next carbon. Next carbon is attached to CH2OH. Here it is attached to OH. Here it is attached to H. And here it is attached to an R1 group. So, we can consider this whole group as R1. Okay. So, that means this is a chiral carbon. All 4 atoms are different. Here all 4 atoms are different. So, that means this carbon is an asymmetric carbon. So, this is a chiral carbon. So, this is correct. Similarly, for this also you see this is attached to this group OH, H and this group. All groups are different. So, this is CHOH, CH2OH. Here we have CHOH, CHOH, CH2OH. So, 3 carbons are there and here 2 carbons are here. So, we have an OH and we have an H. So, all 4 atoms are different. That means this is also an asymmetric carbon. You see this carbon, we have 3 carbons here, 2 carbons here and then H and OH. So, this is also an asymmetric carbon. See this carbon, this is also an asymmetric carbon. We have CH2OH, H, OH and this big R group. So, that means this is also an asymmetric carbon. See this carbon, this has 2 hydrogens attached. This is also CH2OH. So, C, 2 hydrogen OH and a R group. So that means 2 of the 4 molecules are same. So this is not a chiral carbon. So how many carbons are chiral? 1, 2, 3 and 4. So glucose has 4 carbons that are chiral. Now coming to fructose. Now see, for fructose you have this first carbon as not non-chiral, because again 2 of the 4 atoms are same. So this is not chiral. So this won't be considered. Now this carbon, fructose is a ketohexose, so it does not have an aldehyde group. What does it have? It has a ketose group. Wait, this glucose structure is wrong. This will be CHO. This won't be CH2OH. This will be CHO. Okay, and still this carbon will be asymmetric because it will be C double bond O H and R group. So this has only 3 atoms attached. So 2 of the 2 bonds are attached to oxygen. So this will not be considered as chiral carbon. Similarly, for this carbon also, this carbon is attached to C double bond O, CH2OH and an R group. So 2 of the 4 bonds are attached to oxygen. So this will not be considered as chiral carbon. Now these 3 carbons are chiral. You can see this is a different group, this is a different group, this OH and this H, all 4 bonds are different. So this is a chiral carbon, this is a chiral carbon and this is a chiral carbon. Again this is a different group, hydrogen, hydroxyl and then CH2OH. So fructose has 3 chiral carbons, whereas glucose has 4 chiral carbons. So do you understand how chiral carbons are counted? Now if I give you a molecule, a random molecule, if I have CHO, COH, H, I am just making a molecule randomly. Tell me how many carbons are chiral. So this is not a chiral carbon. We will take another ink color. So first carbon is not a chiral carbon. Okay. This second carbon, is this chiral? We have one CHO group, H, OH and this big R group. So this is a chiral carbon. So this is a chiral carbon. Now see this second carbon, it has C, CHO, HOH. Here it has C, CH2OH, CHOH. Here we have OH, here we have H. That means this is also a chiral carbon. This carbon is also a chiral carbon because this group is different, this group is different, this group is different and this. This is not a chiral carbon because it has CH2, 2H are there, so it won't be. So again this molecule also has 3 chiral carbons. Okay. Okay, so you understand how we are counting the chiral carbons, right? Okay, so Akshara has answered 3 already, so very good. So you understand how we are seeing whether it has an asymmetric carbon or not. Okay. Now the next question. The total number of stereoisomers possible for an aldohexose is. So what would be an aldohexose? Aldohexose means it has aldehyde group and 6 carbons, right? So 1, 2, 3, 4, 5 and 6. Okay, this is an aldohexose. How many stereoisomers are possible? So I have told you stereoisomers are based on the number of chiral carbon. Okay, stereoisomers are based on number of chiral carbon. Why? When we will have an asymmetric carbon, so let's say a carbon is attached to 4 different group, R1, R2, R3 and R4. Then only we can have, let's say from this R1 and R4 are fixed. So if I have to see between R2 and R3, we can have two possible conformations that are mirror image. We can either have this and we can either have R2 here, R3 here and R4 here, right? Right, in a chain. Now this can't be changed, this is a straight chain, right? So R1 and R4 are fixed. The difference can be between R2 and R3. So for each chiral carbon we have two isomers possible, okay, two isomers for one chiral center, okay, one chiral carbon. So how many chiral carbons are there in aldohexoses? How many chiral carbons are there in aldohexose? So this is an aldohexose here. I'll erase this. So this is an aldohexose here. Tell me how many chiral carbons. I have just taught you how to count the chiral carbons. So tell me. So this is not a chiral carbon, since it is double bonded to oxygen. This is not a chiral carbon. This is a chiral carbon, right? Why? Because it is attached to CHO, H, OH and this R group. This is a chiral carbon because it is attached to 4 different groups. Similarly, this is a chiral carbon and this is a chiral carbon. So it has 4 chiral carbons, right? So if we have 4 chiral carbons, that means each chiral carbon is giving rise to 2 isomers. Okay, one carbon is giving rise to 2 isomers. That means number of stereoisomers, will be equal to 2 raised to the power of n, where n is the number of chiral carbons. So n is equal to 4 here. So that means 2 raised to the power of 4. So what would be the answer? 16. Okay, the correct answer is 16. Okay, do you understand? We have 4 chiral carbons. Number of isomer is 2 raised to the power of n. That means 2 raised to the power of 4, that is 16. So correct answer is 16. For a aldohexose, number of stereoisomers possible is 16. Similarly, if I ask you, now if I erase everything, if I ask you to tell me the number of possible stereoisomer in aldotriose. So aldotriose will be CHO, C, OH, H and CH2OH. This is aldotriose. And what is the name of this aldotriose? I have told you earlier, this is glyceraldehyde, right? This is glyceraldehyde. We have studied this is the simplest form of carbohydrate, glyceraldehyde. This is an aldotriose. Now tell me how many chiral centers do you see in this aldotriose? How many chiral carbons are there in this aldotriose? Yeah, Akshara has answered 1. Where is this? This is the chiral carbon. Okay, this is not a chiral carbon, this is not a chiral carbon. The only chiral carbon is this carbon. Now, if it has only 1 chiral carbon, that means only 2 stereoisomers are present, because n is equal to 1. That means number of isomers will be equal to 2 raised to the power of 1, that is 2. So what would be the second stereoisomer? It will be a mirror image of this, that is CHO, COH, H and CH2OH. Okay. So you see, this is a mirror image of this. These two are also enantiomers. You can write this as D aldotriose and L aldotriose. Okay. Why? Because this chiral center. Here the hydroxyl group is on right hand side. So this is D-glyceraldehyde and this is L-glyceraldehyde. Do you understand how we are determining the D and L nomenclature and how we are counting the number of stereoisomers? So everything is based on the understanding of chirality. So if your molecule is chiral and if your molecule has more than one chiral center, so based on that you will have to calculate the total number of stereoisomer. Okay, so if it has 2 chiral centers, it will have 4 stereoisomers. If it has 3 chiral centers, it will have 8 stereoisomers, 2 raised to the power of n. So 2 raised to the power of the number of chiral carbons that are there in the molecule. Okay, this is clear? This is clear, right? Okay. Okay, moving to this question number 20. Identify the bond labeled in the structure. Alpha 1 options are alpha 1,2 glycosidic bond, alpha 1,4 glycosidic bond, alpha beta 1,4 glycosidic bond and beta 1,6 glycosidic bond. So identify the bond labeled here. Again you have to number the carbons and then tell me the name of the bond. Come on, now we have discussed many questions similar to this, so you should be able to answer. Tell me, what is this bond, the labeled bond? What is this bond? Whether it is alpha or beta, and what would be the numbering? So if I start numbering here, 1, 2, 3, 4, 5 and 6. Okay. Now tell me whether it is an alpha glycosidic or beta glycosidic. So we have the oxygen here in the same direction as the 6th CH2OH, 6th carbon, right? So if it is in the same direction on the same face of the ring, that means it is beta. Okay, so it is beta. And now the numbering, so 1, 2, 3, 4, 5 and 6. So here also you see this is also beta and the linkage is between 1 and 4. So beta 1,4 glycosidic linkage is what is shown here, and this molecule here is lactose. Okay, it is a disaccharide of glucose. 2 glucose units joined together to make lactose. Okay, this is clear. So this is beta 1,4 linkage. Okay, beta 1,4 linkage. So correct answer is option C. Okay. Which of the following has only one isomer? Now I have told you how to calculate the stereoisomers. Okay. So which of the following will have only one isomers? One isomer means how many chiral carbon? How many chiral carbon are there for a molecule to have one isomer? Number of chiral carbons are 0, that means, right? 2 raised to the power of 0 would be equal to 1. Okay. So we have aldotriose, aldohexose, ketotriose and ketotetrose. So we will draw each and we will see. Or I have the structure. So yeah. So this is aldotetrose. I'll draw the aldo trios for you. So this is CHO, COH, H and CH2OH. So we have aldo trios, aldo tetros, aldo pentos and aldo hexos here. Okay. Now you see the number of chiral carbon in these aldos is the number of chiral carbon in these aldos is, this aldo trios has 1 chiral carbon. This is asymmetric, so 1 chiral carbon. This aldo tetros has 2 chiral carbons. Aldo pentos has 3 chiral carbons. And aldo hexos has 4 chiral carbons. Right? So number of stereoisomers, just a second. Just a second. Yeah, is my screen visible? So aldo trios has one chiral carbon. That means number of isomer will be 2 raised to the power of 1, that is 2. Here the number of isomer will be 2 square, that is 4. Here it will be 2 raised to the power of 3, that is 8. Here it will be 2 raised to the power of 4, that is 16. Now come to ketoses. Now ketoses, this is the simplest ketose that has 3 carbon, 1, 2 and 3, right? Now you see, is any of the carbon chiral? So if you see here, this carbon is not chiral because it has 2 hydrogens. This carbon is not chiral because it is double bonded to an oxygen. This carbon is also not chiral because it has 2 hydrogens. So keto trios has 0 chiral carbons. Now let's see. So keto trios has 0 chiral carbons. Okay. 0 chiral carbons, that means the number of stereoisomer would be 1. There won't be any other molecule. There won't be a mirror image to this, or there won't be a isomer to this. Okay, the only structure possible is this. Okay, for these all structures there are, for all these molecules there are, for this one 2 structures are possible, for this one 4 structures are possible, for this one 8 structures are possible, and so on. For this molecule, let's see, 1, 2, 3 and 4. This is called ketotetros. Okay, 4 carbons, so ketotetros. This is not a chiral carbon, this is not a chiral carbon. It has only 1 chiral carbon. And which one is it? It has this carbon as the chiral center. So it has 1 chiral center, that means 2 stereoisomer. Similarly, this one has 2 chiral centers, so 4 stereoisomers. This one has 3 chiral centers, so 8 stereoisomers. Okay. So this is clear. So this is keto trios. This is keto tetros. This is keto pentos, and this is keto hexose. This is fructose basically. Okay, keto hexose. This is glucose. Okay. So this is clear to everyone. So this was the last question of this session. So you have understood the concept of chiral carbon, the concept of stereoisomers, what is the difference between epimer, anomer and enantiomer, how do we write D and L nomenclature, how do we write the alpha beta nomenclature, how is the cyclization happening in a glucose molecule? The cyclization is happening between the aldehyde and the alcohol group. Okay, what is it forming? It is forming an hemiacetal. Then the linkage between two sugar molecules, that is the glycosidic linkage. So how do we determine whether it is an alpha glycosidic or a beta glycosidic linkage, and between what two carbons the linkage is there? According to that we will have the nomenclature. So it will be alpha 1,4, beta 1,4, beta 1,6, alpha 1,6, similar, and so on. So these are some key points that you have to remember in this chapter. What are aldoses, what are ketoses, like whether it has an aldehyde group or a keto group. Then what is the test we do to detect reducing sugars? How are we doing that test? What is the detection point? The detection point is basically the reduction of Cu2 plus to Cu plus. That Cu plus basically that makes Cu2O, which gives a red color, and it is detected, like it is visible. So we can say that okay, the given sugar was reducing sugar. Okay, so these are some key points. If you have any doubt in any of the points that we have discussed, in any of the questions that we have discussed, please ask. So this was all about carbohydrates. From the next session onwards we will start bioenergetics. Till now we have completed all the biomolecules: amino acids, proteins, nucleic acids, carbohydrates, lipids, vitamins. So we have completed all the major biomolecules. Now we will see how these biomolecules are going to be used in our body for generating energy. Okay, any doubt? So if we don't have any doubt, we can close this session now. Thank you everyone for joining. I'll stop the recording now. Thank you.